Hello friends, I am here with my channel Solving Questions with Gauri ma'am. Today let's go through one question from TIFR GS examination. The alpha helix in protein is formed due to, the options are intramolecular hydrogen bonds, intermolecular hydrogen bonds, or it is due to van der Waals interactions between amino acid or ionic interactions. So let's have a look at these interactions and understand what is alpha helix to find the answer for the question. Yes, if we observe the polypeptide chain which is forming alpha helix, we find there intramolecular hydrogen bonds. These are formed between the first amino acid and the fourth residue apart from that amino acid. So here the C alpha carbon are shown, and this is a first amino acid N, this is N plus 1, then N plus 2, N plus 3, N plus 4, N plus 5. So here the carboxyl CO group of first amino acid forms a intramolecular hydrogen bond with the NH group of the fourth amino acid. This way, there are intramolecular hydrogen bonds present in a alpha helix. Here, 3.6 amino acids are present per turn in a alpha helix. And in between this 3.6 amino acid, there are 13 atoms are present. So that's why the alpha helix is also known as a 3.613 helix. It is giving a helical twisted structure in a protein. When we look at the beta pleated sheets, we find that two extended polypeptide chains are present. This is one and this is another. So these are aligned with each other and these are held together with intermolecular hydrogen bonds. The same way as a alpha helix, here also CO group of one strand and NH group of another strand is forming a hydrogen bond. This beta pleated sheet can be an anti-parallel beta pleated sheets or it can be a parallel beta pleated sheets. In the parallel beta pleated sheets, both the polypeptides are in a parallel direction, carbon to nitrogen. In case of anti-parallel beta pleated sheets, one is from carbon to nitrogen and another is from nitrogen to carbon. So that is in an anti-parallel manner. Here the anti-parallel beta pleated sheets are more stable as compared to the parallel beta pleated sheets. So here we find the intermolecular hydrogen bonding. Now we'll look at the different levels at the structural levels of the protein. We find that the protein shows primary structure, secondary structure, tertiary and quaternary structures. So in our primary structure there is a sequence of amino acids given that we call it as a primary structure. This get twisted. If this primary structure get twisted, it forms a alpha helical structure or it can form a beta pleated sheet. The secondary structure further make complex structures which we call it as a domain. So it get twisted more and it can form a domain like structure, that is the tertiary structure of the protein. During the formation of tertiary structure, there is requirement of van der Waals interaction in the protein. So here the van der Waals interactions are involved. Also the ionic interactions are there during the formation of tertiary as well as the quaternary structure of the protein. What is quaternary structure? When these once these different domains have formed, now they get assembled with each other and whole unit is called as a quaternary structure of that protein. So we always say that there are are 4 domains present in that particular protein. If we see the MHC in immunology, so we find different domains are present. So what are that domains? That are nothing but the tertiary structure of that protein, and then get assembled to form the quaternary structure. So van der Waals interaction and electrostatic interactions, or we can say ionic interactions, are involved in the formation of this tertiary and the quaternary structures. Yes, with this discussion we find that the answer for the question would be option A, that is the alpha helix in a protein is formed due to intramolecular hydrogen bonds. Intermolecular hydrogen bonds are required for the formation of beta pleated sheets. Van der Waals interaction and ionic interactions are there in the formation of tertiary structure of the protein. So this way the answer is option A. I want to inform you that I have started making lectures also based on the subject from the BSc Biological Sciences. It may be zoology, microbiology, biochemistry, biotechnology, botany. All the students from the life science can have benefit from my lectures. So please keep watching my videos and please like, share and subscribe for my channel. Thank you.